Good afternoon, students. In last class, we discussed pollen grain after gynoecium part of, so that is pistil and embryo sac. So megasporangium, that is the ovule, it is a covered with two wall layers, that is integuments. Below that presence of nucleus, inside that presence of embryo sac. That embryo sac is developed by the cells of nucleus. We discussed in this class. So before that, we discussed the structure of embryo sac. The embryo sac, which consists of two ends, as the two ends of ovule, that is chalazal end, and another one end is micropylar end. Micropylar end. This end presents towards the pore in the ovule where the integuments are absent. This pole or this end which is present towards the broad base of ovule, that is chalaza, that is the chalazal end. So in previous class also we written the diagram. So just we are not going to deeply, just to be introduced. So in this class we discussed a detailed study of this embryo sac. The embryo sac is a female gametophyte. A female gametophyte because it is having an egg cell. So it is the egg cell. Due to presence of egg, that is the female gamete produced by in the embryo sac, as it is called the female gametophyte. So the female gametophyte is always seven-celled nature, seven-celled condition. In that seven-celled and eight-nucleated condition. Seven-celled and eight-nucleated, very important. Which are the seven cells? That is, three antipodal cells, one centrally located, that is central cell, and the three cells towards the micropylar end, that is egg apparatus. Egg apparatus. In that egg apparatus, there is one egg cell. In the egg apparatus, presence of one egg cell and two synergids. As the female gametophyte or the embryo sac is seven-celled nature, these are the chalazal end. There is presence of which are located, that is the chalazal end. These three are present in micropylar end. And the centrally located, that is central cell. So we discussed one by one. Here, in the chalazal end, there is presence of antipodals or antipodal cells. In the center, there is a central cell. Inside the central cell, there is presence of two polar nuclei. Two polar nuclei. In the in the micropylar end, there is presence of two synergids. That is two synergids. On the wall of synergids, there is presence of a number of finger-like projections. The finger-like projections, those finger-like projections are called as filiform apparatus. Filiform apparatus. On the synergids, they are attached with a cell, an important cell, that is what they are called as egg cell or egg. So here, the egg cell and synergids, which are called as egg apparatus. Egg apparatus. So just we discussed what are these. In the micropylar end, there is presence of two important synergids. So these synergids are, there is little bit the triangular shaped structures. So these triangular shaped structures which are located towards the micropylar end. In these, there is presence of large vacuoles. And even there is presence of the nuclei. So here the walls of synergids which are little bit thicker towards the micropylar end. Towards the micropylar end, the walls is little bit thicker than towards the chalazal portion, towards the chalazal end. So maybe probably cell walls are thin in nature, but towards the micropylar end, the walls are little bit thick. As the same wall which is attached to the egg cell. So these two synergids which attach towards the basal part, but in the bone center. So as in the back side, like here, the walls of these, the egg cell is attached. So here there is present back side. So there is the egg cell. So it is egg or egg cell. Same, the egg cell walls are also, which is thicker towards the micropylar end. Thicker towards the micropylar end and little bit thinner towards the chalazal part. As on the synergids, for example, just we write a synergid. On the synergid, there is presence of a number of finger-like projections. The finger-like projections. These finger-like projections are called as filiform apparatus. These filiform apparatus, which originated from outer wall, for example, it is the synergid which are originate like this. Sometimes the projection, the finger-like projections which ends, those ends which may be introduced into the synergid. So like these synergids which entered into the cytoplasm of, that is the synergid. The filiform apparatus originate on walls of synergid. Those which projected on the cell walls or on the cell membrane, but those projections which may be entered inside the cytoplasm of synergid cells. So sometimes these synergids which are variate sometimes the ends may be either sharp or blunt end, or otherwise it is little bit that is flat. If any, they may be entered. ಎಲ್ಲ ಫಿಲಿಫಾರ್ಮ್ ಆಪರೇಟರ್ಸ್ ಒಳಗಡೆನೆ ಹೋಗಬೇಕು ಅಂತ ಏನಿಲ್ಲ, ಕೆಲವು ಹೋಗಿರ್ಬೋದು, ಕೆಲವು ಹೋಗದೇ ಇರಬಹುದು. ಬಟ್ ದೋಸ್ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಆನ್ ದ ವಾಲ್ಸ್ ಆಫ್ ಸಿನರ್ಜಿಟ್. ಸೊ ಇದು ವಾಲ್ಸ್ ಅಂದಾಗ, ಸೊ ವಾಲ್ ಮೇಲೆ ಕೂಡ ಇರುತ್ತೆ. What is the function of these synergy? Sorry, these filiform operators. See here, the filiform operators which guide or which show the path, which guide the pollen tube, at the time of which entered into the embryo sac during fertilization. So at the time of fertilization, the pollen tube which brought the two male gametes, and even there is the nucleus of vegetative nucleus. So while the pollen tube entered through the micropylar end, that pollen tube which will enter into the embryo sac, the embryo sac ಒಳಗಡೆ ಹೋಗಬೇಕಾದ್ರೆ, so these filiform operators which show the path, ದಾರಿ ಮಾಡಿಕೊಡುತ್ತೆ, ದಾರಿ ತೋರಿಸುತ್ತೆ, which guiding the pollen tube where it is easily entered into the synergic. Where it is easily entered into the embryo sac. As like these filiform operators which guide the pollen tube, which helps the pollen tube, which show the path to enter the pollen tube into embryo sac. As it helps like a guide. Like the synergics, there are two synergics are there. These synergics at the time of pollen tube entered, where the pollen tube entered into the embryo sac. For example, while the pollen tube entered here, this entire synergid may be degenerate. The entire synergid will degenerate. So means that synergids which helps to easily enter into the pollen tube is easily entered into the embryo sac. So otherwise, if entered here, this synergid will be degenerate. So it is the degenerated one which is shown in the markings. Like the synergids which are degenerate after the pollen tube entered into the embryo sac. So as it has the egg apparatus in which two synergids and one egg cell, this egg cell is also an egg apparatus. The egg cell present just behind these two synergids. It is also, as we shown, that is the wall is little bit thick in micropylar ends, and it is towards the chalaza, it is little bit thin. So the egg cell, it fused with the first male gamete. The pollen tube carried two male gametes. The first male gamete, it is the first male gamete, it is the second male gamete. The first male gamete, it fuses with the nucleus of egg cell, as it leads to formation of zygote. So the zygote formed by fusion of first male gamete with the nucleus of egg cell, as it leads to zygote. That zygote in future is developed as embryo. In future, it is developed as embryo. As these are the three cells which present towards the micropylar end. So the cell which is present in the center, that is a central cell. Those central cells which consist of two nuclei. That is what we are called as polar nuclei. Why it is called polar nuclei? At the time of formation of this embryo sac, one of the nuclei from chalaza end, one of the nuclei from polar, that is micropylar end. These two nuclei which that is migrated from these two poles. One nuclei migrate from the chalaza, another nuclei migrate from the micropylar end, as it is what we are called as polar nuclei. So as these polar nuclei present, that is two polar nuclei present in the central cell. The central cell and even the synergids and that is the egg cell, that is egg apparatus, which covered by a common membrane. So it is a common membrane, it is enclosed. So this wall is also, the wall of this common membrane is also, it is little bit thick towards the micropylar, and it is little bit thin. Here the thin in nature towards the chalaza, but the walls of these common wall of egg apparatus and these central cell is little bit thicker, very thick, little bit thicker than towards the chalaza. So these are the central cell. So another three cells, that is antipodals. So the antipodals, there are three antipodals are there, but sometimes the number of antipodals is different from species to species. For example, in maize there is presence of 20 antipodals. That is geomase. Geomase, it consists, that is the number of antipodals is 20. So, but here commonly each embryo sac consists the three antipodals which are located towards the chalaza end. So there is no role of widely taken commonly, there is no role of these antipodals, but a very important role also is maintained at the time of these antipodals are resistant nature, or that is remaining nature. If the antipodals are resistant, which are present inside the embryo sac even after fertilization, or even after completely matured. See here, at the time of fertilization, while the pollen tube entered into the embryo sac, it releases two male gametes. One male gamete fuses with egg cell, the second male gamete fuses with these, that is the central cell. So that central cell is the mother cell of endosperm kuda karibunnam. Mother cell of endosperm. Ekendra, at the time of fertilization, the second male gamete fuses with egg cell. It leads to formation of pen. That is, pen is nothing but primary endosperm nucleus. the time of fertilization, the second male gamete fuses with the central cell. It leads to formation of PEN, that is primary endosperm nucleus. So at the time of these fertilization, especially here, there is fusing of two male gametes with egg cell and polar nuclei, two times fertilized. That is what they are called as double fertilization, in which first male gamete fuses with these, it is called syngamy. Second male gamete fuses with central cell, it is called triple fusion. That syngamy and triple fusion, it is called as double fertilization. We discussed the next. So as the central cell, it is acts as the source of or the mother cell of endosperm. After fertilization, these three antipodals are degenerate. Or otherwise, here at the time of degeneration of these three antipodals, if they are degenerate during soon after fertilization or before fertilization, these three antipodals are degenerate. They are disintegrate. They are missed. So if sometime, which are the embryo sac which consists the three antipodals which are remaining even after fertilization. After fertilization, these three antipodals may be remaining. During which these antipodal cells are little bit multinucleate condition. Multinucleate condition. These which are the antipodals are remaining, those are little bit larger. They expand in nature. So because multinucleate condition and also which consists a dense cytoplasm and even which consists lipids, proteins, polysaccharides, like that is more the nutrients which are present in each embryo, each antipodals. So these antipodals are nutritive in function when the antipodals are remaining in nature. It's very important. Commonly, antipodals are degenerate, disappear. When they are disappear, before fertilization, they are disappear. So before fertilization disappear agalilla time. So they are after soon after fertilization of takshana degenerate agibidutte. How degenerate agalilla antandre, so which helps to nutritive in function or nutrition. So for which cells they provide nutrition. If the antipodals are remaining, they nutritive to embryo sac. That is the nutritive cells. When they are nutritive, it's very important. So when they are nutrient, the antipodals are remaining even after fertilization. Fertilization ಆದಮೇಲೆ ಕೂಡ ಹಾಗೆ ಇದೆ ಅಂತಂದ್ರೆ, so nutritive function ಆಗಿ role ತಗೊಳುತ್ತೆ. So most of the angiosperms, they are disintegrated, they are disappeared. So as it is just the detailed structure of that is the embryo sac. So how these embryo sac is developed in the that is what nucellus. So because what we say is these embryo sac is developed inside the nucellus cells. So as previously we discussed, there is the structure of ovule. In the structure of ovule, So it is the ovule. So we did not find the embryo sac. So how the embryo sac is developed? So it is a part of nucellus. It is a part of nucellus. So the nucellus, which consists a number of cells, so these the nucellus cells are nutritive in function. So one of the cell, which is the little bit larger. So these are the cells of nucellus. In the nucellus cells, you identified one of the cell is very large, while you compare to all other cells. So these larger cell, we call it as archisporial cell. It is also called as archisporial cell. In certain plants, these archisporial cell, it acts as directly MMC, that is megaspore mother cell, which produce an embryo sac. In most of these cells, in most of plants, these archisporial cell, don't confuse here. One of the cell, ಯಾವ cell ದೊಡ್ಡದಾಗಿರುತ್ತೋ ಈ nuclear cells ಅಲ್ಲಿ, that cell is little bit hypodermal function. So like there is presence of hypodermally, that is nothing but in the nuclear cell, outer covering cells are there. Below that the cell present a larger cell. ಯಾವ cell ನಾವು ದೊಡ್ಡದು ಅನಿಸ್ತಾ ಇದೆಯೋ ಆ ಸೆಲ್ ಮಾತ್ರ ಆರ್ಕಿಸ್ಪೋರಿಯಲ್ ಅಥವಾ ಮುಂದೆ ಮೆಗಾಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಆಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ. So here the nucleus cells, so it is what the developmental stages of embryo sac. So very important for these, for the view of exam or even in the NEET or CET. So how the development of embryo sac, that is the developmental stages of embryo sac. So one of the cell which is identified larger, it acts as a archisporial cell or MMC or megaspore mother cell. So these cells, for example, it is the archisporial cell. These cells which divide two cells so as it divide two cells, the outer primary parietal cell, inner primary sporulous cell. So these outer parietal cell and inner primary sporulous cell, these sporulous cell which take part in production of embryo sac. After some time, it will be degenerate. So as the remaining of these cell, that is it is the, just we write the cells of nucleus nuclear cells. So here these primary parietal cell is degenerate. It is degenerate. It is only remaining. So at the time of here, it will be completely degenerate after some time. So there is remaining only one cell, that is the primary sporogenous cell. These primary sporogenous cell, it divide two cells. Once again, divide two cells. That is the condition of diode nature. After some time, these two cells are divided. Once again, that is the so what the sporogenous cell, which remaining, that is divided into two cells, it leads to formation of diode or two megaspores. These two megaspores which divide once again either horizontally or either vertically. So if they divide vertically, there is presence of 4 megaspores. 4 megaspores. So as like these 4 megaspores present in the nucleus, the arrangement of these megaspores is various nature. For example, it is the nature of isohedral, the arrangement of these 4 cells. Sometimes it will be divided like this, the four megaspores which are arranged like, that is the isohedral nature. Either here, what their arrangement, that is linear nature, linear arrangement, each megaspore which present one above the other. So otherwise, these which are arranged like this, it looks like T-shaped nature. The shape of these 4 megaspores which are arranged in this manner, it is called as T-shaped. As those 4 like these, that is decussate manner. So like different types, these 4 megaspores are arranged in different arrangements. So like we are identified, so different types of arrangements of these 4 megaspores. Okay, so we need not these just we say for improve our knowledge. So like isohedral nature, or linear shaped, or T-shaped, or decussate shaped, these 4 megaspores are arranged. So here, after in the nucleus, what are 4 megaspores which are present in the 4 megaspores present in the nucleus? So it is the nucleus cells. So here, where there is presence of 4 megaspores, like it is micropylar end, it is chalazal end. Just we write here micropylar end, it is chalazal end. Here only one of the megaspore. So this megaspore is remaining. It is the functional megaspore. Functional megaspore. It is a functional megaspore. These three are degenerating megaspores. After some time, after what they divided these two cells, it leads to formation of 4 cells. Out of these 4 megaspores, the megaspore present towards the chalazal end in chalazal ends, towards the chalazal end, the megaspore is remaining. These 3 are degenerate. These 3 are degenerate, as only one megaspore, it acts as a functional megaspore. It acts as a functional megaspore mother cell. Functional megaspore mother cell means ಈ ಒಂದು ಸೆಲ್ ಮಾತ್ರ ಕಂಪ್ಲೀಟ್ ಎಂಬ್ರಿಯೋಸಾಕ್ ಡೆವಲಪ್ಮೆಂಟ್ ಅಲ್ಲಿ ಇನ್ವಾಲ್ವ್ ಆಗುತ್ತೆ. ಈ 3 cells they are degenerate, they are disappeared. So here based on the megaspore which are produced by either single, either 2 cells, either all 4 cells. In most of the angiospermic plants, the development of embryo sac donated by a single cell, this type of development is called as monosporic development. Monosporic development of embryo sac. So mono, it is nothing but the embryo sac. So entire embryo sac, it is produced by, it is donated by only one functional megaspore. So that is what we are called as monosporic development. Sometimes here what we know, out of 4 megaspores, the megaspore present towards the chalazal end is remain. These 3 are degenerate. The megaspore remaining towards the chalazal end, it provides the complete embryo sac. That type of embryo sac development is polygonum type. It is the polygonum type of so like a plant named polygonum. So like here, the type of embryo sac development is polygonum type. In polygonum type, it is also monosporic development in which one of the megaspore present towards the chalazal end. But is another one type is there, but just we see keep it in your mind. So these cell is also degenerate. The cell which is present micropylar end, it is the remaining. ಈ ಸೆಲ್ ರಿಮೈನಿಂಗ್ ಆಗಿ ಈ 3 cells degenerate ಆಗುತ್ತೆ. ಈ cell means the cell which is present towards the micropylar end, which helps to production or that is complete synthesis of embryo sac. The type what we are called as Enothera type of Enothera type of development ಅಂತ ಕರೀತೀವಿ. So polygonum type ಕೂಡ monosporic development of embryo sac. Enothera type of embryo sac development is also monosporic. Here in polygonum type only, that is degenerating megaspores. It is functional megaspore. But in unotherapy, there is one of the megaspore present remaining towards the micropylar end. Micropylar end kade irtakkanta megaspore remaining agi complete embryo sac na development ಮಾಡುತ್ತೆ. Chalazal kade irtakkanta 3 megaspores degenerate ಆಗುತ್ತೆ. Like it is also the monosporic development. So another type of development of embryo sac is bisporic. Bisporic development of embryo sac. So as the name indicates, the two spores, that is the two spores in which in bisporic development in which the two cells present towards the micropylar end is maybe the two cells remaining, the two cells which remaining towards the chalazal end, the micropylar is degenerate. So it is the type of oleum type. Oleum type. In oleum type of bisporic development, so ಇಲ್ಲಿ 4 cells ಇದೆಯಲ್ವಾ, ಈ 4 cells ಅಲ್ಲಿ ಕೆಳಗಡೆ 2 cells ಉಳ್ಕೊಳ್ಳುತ್ತೆ, ಮೇಲ್ಗಡೆ 2 cells degenerate ಆಗುತ್ತೆ. The cells which are remaining towards the chalazal are, that is act as a functional megaspore. The two are, that is degenerate. So that is the type what we are called as oleum type of embryo sac development. So another one is there, that is endymion type. So the endymion type of embryo sac development, it is also type of bisporic development. So these in endymion type, there is which are the cells which are present towards the micropylar end is remain, and the two megaspores present towards the chalazal end are disappear. So this type of embryo sac development is also called bisporic, that is otherwise oleum or endymion type, depend upon the cells present which side, either chalazal or micropylar. So based on even the two cells are also provided to complete production of the embryo sac. So another one is called as tetrasporic development. Tetrasporic development of embryo sac. Tetra, it is nothing but all 4 functional megaspores. All 4 functional megaspores. ಅಂದ್ರೆ ಇಲ್ಲಿರ್ತಕ್ಕಂಥ ಎಲ್ಲಾ 4 cells ಕೂಡ which able to produce complete embryo sac. ಒಂದು embryo sac formation ಆಗಬೇಕು ಅಂದ್ರೆ ಎಲ್ಲದರಲ್ಲೂ ಇರುತ್ತೆ. So that is what we are called as tetrasporic development. So these tetrasporic development, there is presence of different types: Odoxa type, Pinia type, Plumbago type, that is Pipromia type, Drusa type, Fritillaria type, Plumbagella type. So like different number of types are there. Number of types are there in which. So here also there is all 4 spores, but the number of the cells present in embryo sac is more in tetrasporic development, because all the these 4 megaspores which are provided, which are divided number of times. So as it produced, that is the number of cells in the megaspores, number of cells in the embryo sac. So like there is different types of embryo sac development, that is depend upon the megaspores remaining. Different types of arrangements, what they are arranged in the nuclear cells, by the yeast artisporial cell. Here, so what we have discussed that is one functional metaspore. These three are degenerate. So that is we discussed only now. That is monosporic development. Monosporic development. It is only important for our according syllabus. So that monosporic development, it is nothing but the complete embryo sac. It is developed by only one functional megaspore. So these one functional megaspore, it is functional megaspore. See here, the primary sporulous cell which divide meiotically. The primary sporulous cell divide meiotically to produce four haploid condition. So, but here the functional megaspore, it divide mitotically. The nucleus of functional megaspore which divide mitotically to produce two nuclei, means which divide to produce two nuclei. These two nuclei which move towards the opposite poles. See, one thing you remind here, when the nuclei is divided mitotically, it is not followed by the formation of crosswalk. It crosswalk formation agta illa. Andre, ee crosswalk formation agidre, ಎರಡು cells of convert ಆಗ್ತಿತ್ತು. ಆದರೆ ಈ crosswalk formation ಇಲ್ಲದೆ ಇರೋದ್ರಿಂದ both nuclei which divide, each nuclei which migrate towards the polar region. So means here one nuclei moves towards the chalazal, another one nuclei towards the micropylar. So what are the nuclei of functional megaspore divide mitotically to produce two nuclei, each nuclei which move towards the opposite poles without a cross wall. So these nuclei continuously divide one more two mitotic cell divisions. So here, that is what we say is that is it is the end of micropylar end. But this embryo sac is two nucleated embryo sac because it consists of two nuclei. That is two nucleated embryo sac. The nuclei present both ends, which one more two divisions. So there is formation of see here, after these two nuclei formed by these functional megaspore nuclei, the second mitotic division is also not followed by formation of crosswalk. Illu kuda crosswalk formation agaliಲ್ಲ. So as it is formation of that is four nucleated embryo sac. So as it is chalazal end, it is micropylar end. So like one more mitotic cell division, the two nuclei present in the opposite poles, as it leads to formation of there is two nuclei present in each polar region divide mitotically to produce 4 nuclei at each end. That is 8 nucleated. 8 nucleated embryo sac. 8 nucleated embryo sac. See, after formation of 8 nucleated embryo sac, it is suddenly formation of cross wall. ಇಲ್ಲಿ cross wall formation ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಸೊ ಯಾವಾಗ ಫಾರ್ಮೇಷನ್ ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಯಿತು ಕ್ರಾಸ್ ವಾಲ್, so like there is cross walls are formed around the cells. During formation of cross walls, one of the nuclei from chalazar, one of the nuclei from micropylar which migrate into the center. So like here the 3 cells are present here, that is antipodals. What the 3 nuclei present towards the chalazar are the antipodals or antipodal cells, which are the 3 nuclei present towards the, that is micropylar end, which are called as egg apparatus, that is egg cell. So these are synergids, that is egg apparatus. So what here, one of the nuclei from chalazar end, one of the nuclei from micropylar end which migrate, means it leads to fusion. Chalazar end ಕಡೆಯಿಂದ ಒಂದು nuclei, micropylar end ಕಡೆಯಿಂದ ಒಂದು nuclei, they are migrate towards the center. ಅವೆರಡು ಸೆಂಟ್ರಲ್ ಹೋಗಿ ಫ್ಯೂಸ್ ಆಗುತ್ತೆ. ಸೆಂಟ್ರಲ್ ಆಗಿ ಫ್ಯೂಸ್ ಆಗಿ ಅಲ್ಲೊಂದು ಸೆಲ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ. So that is what we call as central cell. Central cell. ಇಲ್ಲಿ we show two nuclei. That is the nuclei what we are called as polar nuclei. Polar nuclei. So like a complete, that is a matured embryo sac is formed. A matured embryo sac is developed by only one functional megaspore. One functional megaspore. So this type of one functional megaspore which provide or which produced, that is two nucleated embryo sac, 4 nucleated embryo sac, 8 nucleated embryo sac. In that 8 nucleated embryo sac, there is presence of 7 cells. Which are the 7 cells? Means 3 antipodals, 1 central cell, 3 egg apparatus. But this embryo sac is, the matured embryo sac is 8 nucleated. Out of that 8 nucleated, 1, 2, 3, 4, 5, 6, 7, 8. As 7-celled 8 nucleated embryo sac is developed by a single functional megaspore, as it is called as monosporic development of embryo sac. So it is the developmental stages of embryo sac by one of the cell, that is the hypodermal cell present in the nucleus. The nucleus cells ಇರತಕ್ಕಂತ ಯಾವುದೋ ಒಂದು cell which divided to produce a complete embryo sac. So ಇಲ್ಲಿ just we go to ploidy level. In the ploidy level, the antipodal cells are all haploid nature. Antipodal cells are haploid nature. The egg cell and synergid cells are also haploid nature. But the central cell, it is diploid. It is 2n number. So why it is diploid means the central cell, it is formed by fusion of two nuclei. One nuclei from chalaza, another nuclei from micropylon. As it leads to formation of a diploid central cell formed in the embryo sac. It is diploid condition as it is one of the mother cell for endosperm, formation of endosperm in the endospermic seeds. So like it is a detailed structure of embryo sac before what we are known. After that, the developmental stages of embryo sac by a monosporic. So already we discussed either monosporic development or bisporic development and tetrasporic development of different types of embryo sac developments. So this is the end of the developmental stages of embryo sac. In next class, we discussed pollination. Thank you.